Welcome to today's event, The Shot Felt Around the World. What's next for COVID-19 vaccines with three world-renowned medical experts, Dr. Katalin Karikó, Dr. Paul Offit, and Dr. Drew Weissman. We are truly honored that they have taken time out of their very busy schedule saving the world so they could join us for this conversation. Thank you in advance for taking the time today. As many of you know, several of the COVID-19 vaccines rely on mRNA technology, and our first two panelists are the leading minds behind the science. Dr. Katalin Karikó has been Senior Vice President at BioNTech and mRNA Pharmaceuticals since 2013, and she is also Adjunct Associate Professor at the Perelman School of Medicine at Penn, where she worked for 24 years. Her groundbreaking research on RNA has led to 10 US patents and several awards. Her patent co-invented with Drew Weissman on a nucleoside modified uridines and mRNA is used to create the vaccines used by both Pfizer and Moderna. Her co-inventor, Dr. Drew Weissman, is a professor of medicine at the University of Pennsylvania's Perelman School of Medicine, where his laboratory focuses on the study of RNA and innate immune system biology and the application of these findings to vaccine research and gene therapy. And finally, we are delighted to have Dr. Paul Offit back with us today. He is the director of the Vaccine Education Center at the Children's Hospital of Philadelphia, as well as the Hilleman Professor of Vaccinology and Professor of Pediatrics at the Perelman School of Medicine at Penn. He's the co-inventor of a rotavirus vaccine that's been credited with saving hundreds of lives every day. It is incredible to have this amazing set of Penn faculty members and inventors here to educate all of us about the vaccines surrounding COVID-19, when we might be able to expect to get one and what's coming next. So let me turn the floor over to Dr. Karikó to kick us off.
Thank you. So we scientists mostly like to look forward, to look ahead, look at the future, to figure out what is next. But first I thought that I would say something about the past, how the mRNA became therapy. And messenger RNA was discovered 60 years ago, 1961. And most of the development was in my lifetime, although 1961, I still was 6 years old and not reading Nature, where it was published, that some unstable RNA was discovered, which is taking the message out of the nuclei from the chromosome to the cytoplasm where translation occurs. We have to wait like 20 more years when we could first make RNA in a tube at the bench, which is critical right now to think about the mRNA vaccine. But I want everybody just think for a second that a year ago, message came over from the internet and we could learn what is the sequence of the virus. And if it would happen 20 years ago, you had to have physically the sequence in your hand, so the viral construct in your hand. But here the information was sufficient. So a lot of scientists worked on it to make it happen because we needed PCR, we needed sequencing that our Chinese colleague could sequence the virus. And that's what happened during these years, you know, the advancement of different biological techniques that made it possible, that getting the information, and maybe 2 days later, the DNA, which provided the template for the RNA synthesis, could be in our hands. And making an RNA takes a couple of hours, and voila, so very quickly, we could have a vaccine. Of course, you know, the process of formulation, again, LNP and lipids also took about like 40 years. To develop and improve, remove toxicity, and have something that we can wrap the RNA in and have a vaccine already less than a year to be ready. So there are a lot of advancement, many field of science, a lot of scientists, hundreds of scientists worked on things that they were not even aware that one day it will be useful for the vaccine. So I just want to say that we represent here all of these scientists and people who work very hard to have this vaccine to be ready. Thank you. Maybe Drew, you want to say for the next part?
Okay, so I'll start up with the vaccine. And as Katie pointed out, the technology involved in this has been going on for many years. The technology of the mRNA LNP vaccine has also been studied for many years. People say, oh, I'm afraid of this vaccine because it was invented 10 months ago. It wasn't. This vaccine has been developed. We developed modified mRNA 15 years ago. LNPs were around the same time. This vaccine has been studied in the lab and in clinical trials for many years. What we know about it is that A, it's very safe. The reactions are sore arm, swelling, a little bit of flu-like syndrome. Those reactions are telling us that the vaccine is working. That's the vaccine initiating the immune response, starting the response that will lead to protection. The vaccine is incredibly efficacious. They've shown that in both Moderna and Pfizer, it's 95% effective at blocking symptomatic infection. It's 100% effective at blocking severe infection. So that for a respiratory virus vaccine, those are incredible numbers. The vaccine is safe, the vaccine is effective. Those, to me, are the most important things. Let me just mention that when we think about vaccines in the future, there have been 3 coronavirus epidemics in the past 20 years. SARS, MERS, and now COVID-19. You have to assume there's going to be more. So one thing that we and many others are working on are future vaccines, and vaccines that will protect against the next COVID-19-like virus to switch from bats to humans and potentially start another pandemic. So we've already started this work. It's the future of vaccines once we get through this COVID-19 pandemic. Now let me turn this over to Dr. Offit.
Thanks, Drew. So this is a disadvantage of going third. I think you guys said pretty much everything I was going to say, but that's okay. I'll try and say it a little differently. And so in November of 2019, a bat coronavirus made its debut in the human population. It was billed as a winter respiratory virus that was causing pneumonia. I think over the past year, we've learned it is far more than that. It has the capacity to damage endothelial cells, the cells that line blood vessels, causing vasculitis, which is ironic considering that the virus actually doesn't enter the bloodstream for the most part. It induces your immune response to react against endothelial cells. So it can cause strokes, heart attacks, liver damage, kidney damage, which is not true of typical respiratory viruses. It affects taste and smell, and some neuropathologists have been able to identify the virus in the brain as it travels up from the nose. Again, not typical of respiratory viruses. It has a predilection for people who are older. About 92% of the deaths have occurred in people over 55 years of age. It causes an unusual disease in children, this multi-system inflammatory disease, which I haven't seen before, caused by any virus. So it's been a surprising virus, both clinically and pathologically. To meet that challenge, what we've done is we've met it with a technology that was developed by Drew and Katie, with which we have no commercial experience. Although Drew's absolutely right that we do have research experience, there is not another mRNA vaccine out there. So I think it was really with bated breath that we watched the development of this vaccine. And as Drew said, both the Pfizer and Moderna products have been remarkably successful, at a level I think no scientist would have predicted a year ago. 95% effective across the board, across different comorbidities, across all ages, including people over 65, including virtually 100% protection against severe disease. I think we have to be humble here, realizing that we learn as we go, and there may be some things that we know a year from now that we wish we knew now, but for the most part, it is a remarkable achievement. And it's just sort of, for me, an honor to be on the same podium, if you will, with Katie and Drew. Thank you.
Well, so let me start with a question for Katie and Drew. And all three of you are playing such a fundamental role in making our world a safer place again after all that's happened. And Dr. Weissman, Dr. Karikó, did you ever think your work would have the kind of global impact it's having today? And when you first heard about this new virus, did you already know what needed to be done? You know, was there a moment where you were like, oh, it's the work we've been doing, that's what's going to make the difference? Or, you know, did it happen gradually?
Telling the truth, you know, when I first heard about the virus, I were not thinking about that it will reach us. I thought that it would be local. That's, you know, and I worked at BioNTech, and our CEO vision was that, you know, he could, his genius, that it is coming, and then acted immediately. So I'm more like a laboratory person, and I was more like focusing on other application, actually, for therapeutic application of the mRNA. Andrew?
So, you know, Katie and I, when we first started this research, we knew once we developed modified RNA that it had enormous potential, that it had uses in vaccines, in therapeutic proteins, in fixing genetic deficiencies, in editing the genome. So we knew that it had just incredible potential, and potential across many different diseases. We were both very surprised by this pandemic. I think the whole world was. But this technology was ideal for this pandemic. You could make mRNA in days. It had been proven in animals to be very effective against many different pathogens. We have seen good safety in clinical trials. It was an ideal technology, and both BioNTech, my lab, Moderna, and other labs started working on vaccines with mRNA immediately after that sequence was released.
That's just incredible. And I want to turn to Paul now. And this is a question we were going to ask anyway, but there's actually already a question in the Q&A about this that says, you know, basically a friend of mine is dubious about taking the vaccine, even after I told him the science so far shows it's safe. I mean, and Paul, I don't know if you remember this, but, you know, you told me back in September, you were basically like, if I say to take the vaccine, then take it. So like, what's the Paul Offit word now on these vaccines, you know, for our audience?
Right. No, I think that any reasonable person could be and should be, frankly, skeptical of anything they put in their body, including vaccines. I think a lot of the language that surrounded this vaccine was a little scary. Operation Warp Speed, you know, who's going to be the first to cross the finish line?
Who doesn't like Star Trek?
the race for a vaccine. So I think that scared people. The fact that it's a novel technology, at least for which there's no commercial experience with another vaccine, was frightening, reasonably, for people. And it's interesting that the more frightening the virus is, the more people will rate a vaccine as being dangerous. So people will consider the Ebola vaccine to be dangerous, or the anthrax vaccine to be more dangerous than, say, viruses or bacteria that affect children, because they see that the virus is more dangerous, so they'll equate that means the vaccine is more dangerous. So I that's all okay. I mean, I think you should be skeptical. I do think, though, that when you see the data, I mean, as we did, I mean, I'm on the FDA's Vaccine Advisory Committee, when we looked at the data from Moderna and Pfizer, looked at the FDA's review of all the clinical work, and so you get these 200-page documents for each of those vaccines, and you look through all the data, you can't help but be compelled by the safety and efficacy of this vaccine. And although I think most people don't worry about efficacy, I think they assume it works. It's the safety that worries them. But remember, if you look historically at vaccines, which can cause serious side effects. I mean, the oral polio vaccine could itself cause polio. The yellow fever vaccine could cause something called viscerotropic disease, which is essentially yellow fever. You know, the squalene adjuvanted flu vaccine used in Europe in 2009 was a rare cause of narcolepsy. But when those problems happen, they do happen within 6 weeks of a dose. So although they may be long-term problems, you do see them fairly quickly. That's why the FDA insisted that there be 2 months of safety data after dose 2. So you could say pre-approval that those vaccines didn't have at least a relatively uncommon severe side effect. But as Maurice Hilleman have said, who's, I think, the father of modern vaccines, I never breathe a sigh of relief until the first 3 million doses are out there. Well, the first 3 million doses are out there. I think we're up to more than 15 million doses out there. And there doesn't seem to be a severe problem. So there might be. I mean, there might be a rare problem that raised its head. But for right now, the answer to the question, when you say is a vaccine safe, which people worry about, what you're really asking in the medical world is, do the benefits clearly and definitively outweigh the risks? And then right now, those risks are all theoretical. And the answer to that question right now is clearly yes. I mean, here's a virus that's killed 400,000 people. That's probably going to kill another 100,000 in the next 6 to 8 weeks. The choice to get a vaccine is an easy one right now.
Very persuasive from my perspective, but I'm an easy sell on this. I want to turn back to Drew and Caitlin and ask about how, as the inventors in some ways of the underlying technology, how you've thought about the public messaging surrounding the safety of an mRNA RNA vaccine. Do you think the public discourse about it has been responsible? Do you have any recommendations? I mean, I'm sure people have already asked you, what recommendations do you have for public health officials, whether in the United States or elsewhere, for how to better explain why this is a safe type of vaccine to take?
So I actually spend most of my evenings and weekends talking to lay groups about the vaccine, talking to them about why they should take it, why it's safe, et cetera. I think people are concerned about a couple things. The first is that it was done very quickly. But what they have to realize is that the reason it was done quickly is because there was a major pandemic and we needed a solution soon. If we had done it slowly, we would have been mocked for doing it slowly. But nothing was cut short. There were no corners cut. The reason it was done quickly is everything was done at the same time, and that's unusual for vaccines. The other thing is that the safety parameters are there. Convincing people to take it is another issue. And what I hear from a lot of my friends who are hesitant is, I want to wait 6 months for everybody else to take it until I know it's safe. Well, we're at that 6 months, or just about at that 6 months point from the time that the phase 3 trial started, and it is safe. As Paul says, it is clearly safe, and people need to take this vaccine.
Caitlin, you want to jump in, or?
Yeah, so, I mean, Drew is the expert. As I mentioned, my most focus was on a therapeutic application of the RNA, but yeah.
So one, just to follow up on that, for maybe whoever wants to jump in, one question that's shown up in the media, and that some of the some of our questioners asked about if there are any racial differences in the data, and with some things that people have seen suggesting larger error bars for African Americans than, say, Caucasians. Should we have any concerns about efficacy in particular populations at this point, or is this sort of fall under the umbrella of there's basically the risks are just theoretical at this point?
I guess I'll start with that. It was very important for there's something called the NIH Active Group, which was put together by Francis Collins. So we were involved heavily early on in helping Pfizer and Moderna design these trials. It was critical that we include the Hispanic and Latinx population, that we include Black and African American populations. It was representative of the American public. So there's about roughly 10% Black or African American, for example, and roughly more than about 24% for the Hispanic, Latinx community, which represents the American population. So, so we can say then comfortably that when we go to these communities and say, look, it's important for you to get this vaccine, it has been tested in your group. Although I can't really think of an example of where one racial or ethnic group doesn't respond to a particular vaccine as well as another. I mean, you see obviously age differences in response, but not so much racial or ethnic differences. So that there has been study, but that is an issue. I think the African American community especially is somewhat hesitant to get this vaccine. I think we have earned their distrust over the years, and now I think we need to find a way to gain it.
Drew or Caitlin, do you want
to? I completely agree. That the bigger error bars are just from smaller numbers. The efficacy is the same across all racial groups. To me, what was even more important is the efficacy is high in aged populations. And for many of our vaccines, that's not true, and that's a major problem. The fact that elderly people are well protected this vaccine, since they're the population that suffers the most from the infection, is incredibly important.
So, Dr. Karikó, we have a question from a student that says, thank you for speaking with us. As a science student at Penn, I'm inspired by your work. How do you feel about being at the forefront of the new, medical frontier of the mRNA vaccine? And let me combine that with another question that sort of asked, where did the idea of making modified mRNA come from?
So, as many of the scientists, as I mentioned myself, I'm more comfortable to be in the lab and reading or doing something than in the limelight. And the idea came actually as in Hungary, I already started to work with RNA. Those were not messenger RNA, those were short RNA. And actually my thesis, I used a modified nucleoside to make a short RNA and use as an antiviral compound. These were 2' 5' link compounds. And later I moved to Temple University, and my Professor Suhadolnik, he wrote the book on modified nucleosides, the textbook. And so I was familiar that different modification exist, and when performed an experiment and seeing that, try to figure out how to make the RNA non-immunogenic, because I wanted to use, as I mentioned, for therapeutic purposes. I, in an experiment on dendritic cells, found that tRNA was not immunogenic. And knowing that tRNA, the transfer RNA, has a high percentage, 25% of modified nucleosides, came the idea that maybe if we modify the nucleosides in the mRNA, then we can make non-immunogenic RNA. And of course, we needed also that it should be translatable. And of course, it was a surprise that if we modify the uridine, then it came the RNA non-immunogenic. And we know already 10 years later, showing the two toll-like receptor 7 and 8 crystal structure, showing that why uridine is the immunogenic component in an RNA, and why modifying it, we learn is so important. And making pseudouridine, replacing the uridine, which is also a naturally occurring nucleoside, and made it not only non-immunogenic, but resulted that from that RNA template, so much more protein could be produced.
So a question, I'm not sure who exactly will want to jump in on this first, but we've gotten several questions in the chat that in some ways relates, Perry Worldhouse's focus is global affairs. And so many of the people on this are not medical experts, but have a great interest in how the world works. And we've had several questions asking about vaccines that other countries are developing. And in particular, the vaccines coming out of China and Russia. I mean, look, I'll be honest, as an American, I'm delighted that, we're in the lead, but what do you know about these vaccines from China and Russia? What are they based on? Similar underlying technology? What's your sense of how safe and effective they might be?
So we're living in an age of science by press release. Often you don't see the data you really want to see. I mean, so for example, Russia has their so-called Sputnik program, where they have a 2-dose vaccine. One is replication defective human adenovirus type 5. The second is replication defective human adenovirus type 26. Within just a few weeks of its being tested in phase 1 trials, Vladimir Putin was on the airways talking about how it was highly effective, even though they hadn't even started phase 3 trials yet. Now, apparently they're doing phase 3 trials and claim that it's effective in the low 90% range, but again, not published. Nonetheless, they are giving the vaccine in Russia. They're sending it to other countries like Venezuela, which are also giving the vaccine. So I don't know. I would love to see the data. Same thing with China. I mean, China has an inactivated vaccine, meaning take the virus, grow it up, purify it, and kill it with a chemical, in much the same way we make the inactivated polio vaccine or the rabies vaccine or the hepatitis A vaccine. Again, they had claimed 79% efficacy. Brazil, which apparently was using the same vaccine, claimed 50% efficacy. So I don't know. I mean, it's not, I'd love to see it published. I mean, it's very frustrating these days, sort of trying to get information you can either in preprints or Twitter, which is where often you go to find out information about the variants. It's a frustrating time, but I'll be curious to hear Drew and Katie's point of view.
I mean, in our current world of science, when you look at it, there really are a limited number of vaccines that are being developed. There are RNA, DNA, live virus, which there are none, inactivated viruses, and viral vectors, mostly adenovirus. And everybody is using one of these platforms. So what the Russians, what the Chinese are developing are these platforms, and they're similar to platforms being developed here. But as Paul says, until we see the data, until we see the hard data peer-reviewed that show what are the adverse events, what is the efficacy, we just don't know.
I have no comment.
So another question we've gotten from a lot of the listeners on here involves the different variants of COVID-19. There's the UK variant that now I'm saying this like I actually understand what these things mean. There, like discussions of different US variants. Does that make a difference from the perspective of the relative effectiveness of these vaccines?
So at the BioNTech, we are constantly making the pseudoviruses when new variants occurs, and those are spike protein incorporated to VSV pseudoviral construct, and then testing out using blood from those people who were vaccinated with our vaccine and test out that whether it is still effective. And as of yesterday was already published that BioNTech Pfizer vaccine was effective against the UK variants. And, it takes time because the South African is the next one that you have to incorporate all of these, you have to clone and you have to incorporate all of those mutation in the new product and generate these kind of pseudo viruses and then perform the experiment. And I mean, it takes some time, but in a week or so, we could have the answer. So it's important, of course, because the people are getting the vaccine here, and then they are worried that, oh, there is a new virus around. And so it is very critical that they would be informed that it is protected against the new variants.
Yeah, add this, if you don't mind. So there are a number of variants. There's the UK variant, the South African variant, there's the Brazilian variant. Now, just in the last day or so, there's a California variant. It's a single-stranded RNA virus. It will mutate, as all single-stranded RNA viruses do. As a general, coronaviruses are slow mutators. But is it possible that this virus could mutate away from the immune response that's induced by the vaccine? That's possible. The most accurate way to prove that is if you have someone who's been fully vaccinated, received 2 doses of vaccine, and they are nonetheless hospitalized with SARS-CoV-2. I think it is important to isolate that virus and see whether or not it has mutated away from vaccine immunity. That's the most important way to do it. I mean, Katie's right that when you look at as Pfizer has done, when Pfizer basically takes the sera it's obtained from people who were immunized twice with its vaccine, and then looks at that UK virus to see whether it's neutralized in vitro, it is. But again, that's a predictor. I mean, you were assuming neutralizing antibodies is a predictor, and I think it is. But I think you need to. The most important thing clinically is to see whether this is happening in the real world. And I do worry about this. And right now, we're depending on the companies to do that. They have a precious little amount of sera. Sometimes these assays require a fair amount of sera to do these neutralization assays. So I think what we should do is have hundreds of people who've gotten 2 doses of either the Pfizer or Moderna vaccine donate their blood so that we can have a large pool of sera from immunized people that then more than one laboratory can look to see whether or not when these variants emerge, whether or not those sera neutralize the virus in the laboratory. There's a lot of study on monoclonal antibodies, which is interesting, but we really care about the so-called polyclonal antibodies that are induced by either natural infection or immunization.
I think what people have to realize, and I don't like calling these mutations. To me, they're adaptations. Probably November 2019, this virus made the jump from bats to humans. It had been in bats for a long time. This is the first time it came into humans that we know of. So what the virus is thinking is, I have to learn how to grow better in humans. That's what viruses do, and that's adaptation. So we expected these mutations to occur, and we expect them to continue occurring. And that's allowing the virus to grow better in humans. The issue and the unknown right now is what Paul just mentioned is, are these mutations avoiding the immune response from either already infected people or from the vaccine? So far, we're not seeing that. The concern is that a paper was recently published on cold virus coronaviruses. So these are common cold viruses that have been around for forever. And what they see is that there are immune evasions. So these viruses will change to avoid immune detection. So to me, that's the most concerning thing about COVID-19, is that we may get to a point where there's enough immunized people or enough infected people that the virus is going to be driven to avoid these immune responses. And we have to respond to that quickly.
That's frightening. Let me ask another question that's come up frequently. It might come up frequently. I'm going to take the privilege here of being the moderator and ask the question that my mother wants to know about, who's also on the event, which is how safe is this? Are these vaccines for children, under 16? Should children be vaccinated, or should our focus be more on vaccinating adults? And then relying on that to manage the risk of children being infected.
So people less than 21 years of age make up about 26% of the US population. However, they account for 0.08% of the deaths. So children were not an initial target for study. They are now. I mean, we are now testing. Jeff Gerber at Children's Hospital of Philadelphia and others are now doing a variety of trials with different vaccines, looking down to actually as young as 2 months of age to see what we can find in terms of the degree to which these vaccines induce what we hope to be protective immune response in children. So these studies are being done. I mean, do I think that vaccines need to be given to children? Yes, because children can suffer with these diseases. Children can be hospitalized with this disease and can occasionally die from it. So, I mean, we have at Children's Hospital of Philadelphia a COVID ward. There are always children in it. And I think if we can find a way to prevent this virus safely in children, then we should do that. So these studies are ongoing. I would imagine by the summer, we'll have very good immunogenicity data and dose ranging data in children. I'm not so sure we're going to see big prospective placebo-controlled efficacy trials, but I think you'll at least see immunological studies in children.
I mean, ultimately, we have to immunize the children, because if we want to achieve herd immunity, that means the kids need to be immunized as well. The kids need to stop spreading the infection. The bridging studies, which are done to move vaccines from adults to children, are being done now. So we should know soon, in the next months, is it safe and is it effective in kids? And then I hope we'll start immunizing our kids.
So let me follow up with another group where there's been several questions in here, which is, what about pregnant women? Should pregnant women be taking the vaccine, or should they be holding back and relying on herd immunity eventually developing?
This is the same, just like with the children, that you have to first collect data. And in not just this trial, any kind of drug trial, pregnant women would be the last one to test out because, all of the safety reasons. So there are, people who are during the clinical trial who sign up and then they became pregnant, and they are following up those and then collecting data to see the consequences. But it's right now, as far as I know, there were no adverse effects. But the numbers are low.
Right, there in the Moderna, with the Pfizer trial, there were 23 women who became pregnant during that trial who received either vaccine or placebo. And the Moderna trial was 13. Again, broke out equally pretty much vaccine and placebo. There were no problems with the women at that time. I don't think they had delivered yet. The CDC is very clear on this. They know that women who are pregnant who require SARS-CoV-2 infection are more likely to suffer a severe infection than women who are of the same age and not pregnant. So therefore, they say that women can reasonably choose to get this vaccine. That's good. Can I ask a question actually, as long as I have, I was lucky enough to have Katie and Drew here. Were there preclinical studies, I assume there were preclinical studies in pregnant animals to see whether or not one, the lipid nanoparticle could cross the placenta, and also is there any evidence in terms of its ability to cross the blood-brain barrier?
So those studies were not done and were not done well. Some of them have been done. As far as pregnant animals, we've immunized pregnant animals, we've immunized animals who have gotten pregnant. The issue is that if you talk to the maternal fetal researchers, what they say happens is that inflammation from an infection, from a vaccine, from anything, but intrauterine infection leads to neurologic damage that in kids may not show up for years. The pregnancy can end completely normally with a healthy baby born, but you don't know the long-term damage. And that's the big concern that we've got. If you look at LNPs, the placenta has ApoE receptors. And what that means is that LNPs bind ApoE, which is a serum protein, and that's how they target the liver. So potentially they could target the placenta. And these studies are ongoing and they need to be analyzed. I just heard that the country of Israel is aggressively immunizing all of their pregnant people because they've had some bad outcomes in pregnant mothers. And I think the bad outcomes will probably outweigh any potential risk, but all of this is unknown. And I'm data driven and it makes me nervous.
As to my knowledge, Pfizer performed experiments and vaccinated rats that were pregnant and follow up, and they couldn't find any adverse effect on those animals when they vaccinated. But to add to your point, what the infection could cause and what would come up later, it was very interesting studies performed after the influenza in 1918 that those who were pregnant and get infected, their children get depression. I remember, and then they show the animal studies also that when they infected, of course, when the animals were double-stranded RNA treated, those the pregnant animals, then their offspring became depressed. So that, what you are making, that maybe the infection has a long-term effect that cannot be seen and, for quite a time. But those were infection and not a vaccination, because in vaccination, you are delivering a minuscule amount of genetic material.
Wow, that was incredible. All right, I want to move from the micro now and thinking about studies a little bit back to the macro, and a question that a few people have asked, which is about global inequality. And this, in some ways, is a philosophy question as much as it is a details of a vaccine, more than it is a details of a vaccine question. But I think there have been a couple different variants of this, but it essentially boils down to global inequality. It's a thing. It's a huge issue. How should we be thinking about what the distribution of effective vaccines should look like when you have countries with different abilities to afford to purchase a vaccine, where if you have limited supply, how should those be distributed between different countries? What do we do about these difficult questions?
So to me, this is a critical issue because The predictions are it'll be years before the entire world is vaccinated. We have these new variants popping up everywhere. So if these new variants are going to keep popping up in parts of the world that aren't vaccinated. And to me, that's a big problem. The issue right now is there's only 2 vaccines approved. When there's 3, 4, 5, 6 vaccines approved, there'll be a lot more to go around and we'll be able to vaccinate much more of the world. But it's important to vaccinate the entire world. Back in March, I was contacted by the Thai government, and they came to me. I've been working with a Thai university for years. They came to me and said, we're concerned that if a vaccine is made in the West, it's going to be years before we ever get it. And we're not willing to wait years. And I've been working with them to make a vaccine in Thailand that will be given to the Thai population in 7 surrounding lower income countries in Southeast Asia. They're setting up the production facilities in Thailand so they can do everything. They can make, distribute their vaccine. And we're hoping to start giving it to people in phase 1 trials soon, and hoping to vaccinate the population by the end of the year.
So just I must say, that inequality, so it would be ideal if the vaccine would be stable at room temperature and would be easy to make tons of it, but and make it very quickly. So there were a lot of demands, and quickly this was only possible that BioNTech Pfizer had the most experience with this -80 LNP mRNA, which we work on for years already. And so it would be, it would be coming the other kind of vaccine, maybe lower temperature or actually higher temperature would be available and then could be more easily distributed. But we can also say that maybe developing countries maybe should buy -80. I don't know that maybe less arm and more -80 freezer, because the importance is that the -80, the vaccines are stable for years. I mean, we had experience with mRNA formulated with the same way, and 6 years ago, we already put it aliquots, and when we are taking out today, it is not different. So, maybe that's the way to keep it longer, because I have to remind you that when it is stored in -20, that's 6 months later, you have to discard the remaining, or those other RNA vaccine for 3 months at room temperature, then you have to discard the remaining. But in the case of -80, you can keep it for years.
So another question that's come up several times in the chat has to do with how often people are going to need to be vaccinated. And that, is this going to be something like the seasonal flu, where this gets back in some ways to the variants question that we talked about before, where every year you're going to need a booster essentially for your COVID vaccine, or is this the kind of thing where people are going to get 2 shots or maybe 1 shot, depending on if other vaccines are developed, and then that's it?
Well, you get a yearly flu vaccine, not because you're just getting a booster. You're getting a yearly flu vaccine because the virus mutates so much from one year to the next. The natural infection or immunization the previous year doesn't protect you. In terms of this particular vaccine, either one of 2 things is going to be true. Either one, that there will be variants that are created that become prominent that aren't being covered by the vaccine. That would be one reason to vaccinate. Again, the other possibility would be that immunity is not long-lived. I mean, I am encouraged by the fact that when you look at the second dose of this vaccine, there are, one detects relatively easily T helper cells, cytotoxic T cells. I mean, that makes me feel comfortable that there's probably a reasonable amount of immunological memory, which should protect you, I would hope, for a few years. But I'll be curious to hear Drew and Katie's comments on that.
I mean, right now data is up to 7 months, and those are looking at those people who were infected. So I think that sometimes some viruses are having tricks to reduce the memory cells, and with the vaccine, I expect that it will last longer. So the durability of the vaccine will be much longer than somebody who get infected.
Yeah, unfortunately, we just don't know these answers, but we're going to know soon. In animal studies, the RNA vaccine gives incredible durability. We're hoping we see the same in people. The issue, of course, as just mentioned, are the variants and whether a variant will develop that we need a new vaccine against. And we'll find out over time.
All right, I have a very specific question for Dr. Offit now that has come up twice, which involves the relationship. I mean, and Drew and Katalin, I suspect you'll have a good answer to this as well. But Paul, apparently you said something in a different forum about taking NSAIDs or acetaminophen in relation to taking the vaccine, and the question of whether that would blunt the immune response. Is that a thing? And as the vaccine becomes more available, how should people be thinking about the taking the vaccine in relation to over-the-counter medications that people maybe take all the time for other reasons?
All right, so when you develop fever, you do that because your immune system works better at a higher temperature. It's why everything that walks, crawls, swims, or flies on the face of this planet can make fever. We do it because it's to our benefit, obviously. There are two studies, one in Australia, one in the Czech Republic, where they looked at people who pre-treated before they got a flu vaccine in one study and before they got a variety of pediatric vaccines in another study, to show that those who took anti-inflammatories or antipyretic, anti-fever drugs, whether that affected the immune response to the vaccine. It did in those cases. So as a consequence of that, the CDC has recommended that people who get these vaccines not take either NSAIDs or antipyretic medicines before they get the vaccine. The data regarding once you've had the vaccine and you start to develop the symptoms, which are not trivial here in terms of low-grade fever or fatigue and stuff. I've already gotten 2 doses of vaccines and definitely had symptoms associated with both doses, but chose to take no medicines because I see this for what it is, which is just my immune system working for me to try and make a response against SARS-CoV-2 spike protein. That's where that recommendation comes from. It comes from those studies.
Drew or Katalin, you want to jump in on this?
I completely agree. I mean, the issue is we don't know. We would have to do studies giving people NSAIDs or not and see if there's a difference in vaccine response. For the most part, we tell patients don't take them. If you have to take something, Tylenol is probably the first drug to take. But until we know, that's all we can say.
I just heard some people taking some anti-allergic medication who are having serious allergy right before getting the vaccine.
So let me ask a follow-up in some ways to the previous question, which is, if we have additional variants of COVID that require a somewhat a different or new vaccine, every year akin to the flu, does that mean that we'll need new processes of clinical trials? Does that mean that the whole infrastructure essentially has to replicate every year, or do the clinical trials up to this point become a platform that are, a basis to ease that process?
I think it is a platform, and this is already true for cancer vaccines, that once we are demonstrating that it is safe and helpful and beneficial, then we don't have to. Especially at BioNTech, we have the individualized cancer vaccine, which each individual will have a different vaccine for their own cancer. And so when you provide for authorities the data, then you do not have to repeat and provide again. So most likely that it will be much easier for next vaccine, mRNA vaccine.
I mean, ultimately, it's up to the FDA, EMA, and other organizations. But the flu vaccine doesn't undergo clinical trials every year. They just stick in the new flu viruses for that year. And I'm hoping that if we ever get to that point, and I hope we don't, that RNA will be similar.
Sorry, Paul, I wasn't sure if you wanted to jump in. Let me, I know we need to get off right at 1. And so let me ask I think the two big questions that I expect a lot of people on here, really want to know the answer to. And the first is, when can we expect to get a vaccine? Similar in some ways to the discourse surrounding testing early on in COVID. There's now this, this question of, there's not enough, we're trying to ramp up production, when will there be enough essentially? And, what does that mean? When should the average person maybe, let's limit this to the US, when should the average person in the United States be, thinking that they might be able to get a vaccine? And I understand this is an unfair question that you don't control the answer to.
I mean, what I would say just briefly is that to some extent, it's going to depend on when other vaccines come onto the market. I think expecting that the 300 million Americans, which for a 2-dose vaccine is 600 million doses, that could all be borne by the RNA technology is a lot to ask. I would imagine that the answer will be, it will be much quicker if these at least 2 other vaccines, J&J's and AstraZeneca's vaccines, come on the market relatively quickly. But I'm curious to hear what Drew and Katie say.
I just agree.
Yeah, I mean, what I heard is that the RNA companies are hoping to be able to produce 1 billion doses by the end of this year. But that's still not enough for the entire world. So we need other vaccines to be approved and used.
So let me ask a follow-up on that I don't know. I mean, I realize I'm getting a little away from your sort of areas of expertise in some ways, but you know a lot more than I do, which is there's been talk that I think this may have even come out in one of the executive orders already, that the Biden administration is going to invoke the Defense Production Act to try to increase the supply of available vaccines. What does that actually mean?
Yeah, so they're nice words. What they're not keeping, or what they're not explaining is that
you're not going to make me feel better now, Drew, are you?
No, I'm sorry, I'm not. That for pharmaceutical drugs and vaccines, they have a GMP production procedure. So that's a very specialized procedure to make drugs. It costs millions of dollars to make a GMP facility. The vaccines, RNA vaccines on this scale are new. We've never had to make 1 billion doses before. So everybody is building up their GMP facilities, increasing their production capacity, and many of these companies are outside of the United States. So the defense production will have no effect on them. Everybody's dependent on everybody else. We need the raw materials to make the vaccines. We need the facilities to make the vaccines. And we need the infrastructure to distribute the vaccines. All of those have to be improved, built up, expanded to be able to increase vaccine production.
All right, well then let me get you all out of here then on the last question is in some ways, shifting a little bit from when will the vaccine be available to, do you remember that before time? Back when we got on airplanes and we went to restaurants and you'd hang out with your friends, not being afraid of them. When do you think, what would be your prediction for when life goes back to normal or a new normal from the perspective of the average American?
I guess I'll start. I think there will be, as we vaccinate more and more, as we move to warmer weather, as we have more vaccines, I think what I imagine would happen is we will see a gradual decrease in the number of cases and hospitalizations and deaths. It's not going to get to 0. I think when we feel comfortable is sort of when there's a certain level of disease that people feel comfortable with. I mean, every year, influenza last year caused 400,000 hospitalizations and 20,000 deaths. The year before that, it was 700,000 hospitalizations and 60,000 deaths. I mean, if we wore masks and social distance, we could dramatically decrease those numbers. We don't. We sort of accept that at some level. And I think This is obviously a newer virus. I think obviously we're scared to death of it, and we should be, but it may get to the point where we're sort of more comfortable with a certain amount of cases and deaths that we feel we can go back to normal. But that's just my guess. Curious to hear what the other guys think.
I completely agree.
Yeah, I hope that it will be this year.
Me too. Well, thank you all for joining us today. Dr. Karikó, Dr. Offit, Dr. Weissman for sharing these insights and taking the time. And thanks to our audience as well. We hope to see all of you next week for our next two events this semester, which are on a very different topic, the future of transatlantic relations, starting with a roundtable of high-level transatlantic leaders on Monday, January 25th, and then a lecture from Professor Andrew Moravchik on global policy and the bark and bite of populist foreign policies on January 26th. As always, you can access a recording of this conversation on our YouTube channel. You can find out about all the great events we have planned by joining our mailing list and following us on Twitter, Facebook, and Instagram at Perry Worldhouse. We'll drop links to all of these things in the chat as well. And thank you again to our amazing panelists. I've learned so much from this, and I hope our audience has as well. We appreciate you all taking the time, and thank you for the incredible difference that you're making in all of our lives.
Thank you.
Have a great day, everybody.